Hello, my name is Kimberly Aguirre, and as you can see from my poster title, my research is on comparing the fecal microbiomes of grazed and non-grazed sheep and seeing if there's an association with foodborne pathogen shedding. So to give a bit of background, this project is looking at mixed crop livestock practices, which use livestock to graze on cover crops or residue before a field gets replanted with new crops. Livestock are known natural reservoirs for foodborne pathogens within the farm environment, making this a major concern within the agricultural industry, as it represents one of the entry points through which foodborne pathogens may enter our food supply. So in order to gain the benefits of this integrated crop livestock system, we want to minimize and prevent contamination of our crops during livestock grazing. And one potential method we can use is animal diet. So some studies have shown that the gut microbiome of an animal can be influenced by its diet, meaning that the presence of pathogenic bacteria in the gut microbiome can also potentially be influenced by diet. So what I'm doing is looking specifically at sheep to see if changing their diet, so going from pre-grazing to post-grazing, will also influence the presence and prevalence of certain pathogens, particularly Shiga toxin producing E. coli or STEC. And I wanted to look at STEC particularly because of its relatively high prevalence in livestock fecal samples, as well as it being a foodborne pathogen that causes significant illness in humans. So I hypothesized that the change in diet would result in both microbiome changes between grazed and non-grazed sheep and an increase in the prevalence of STEC being shed by these sheep. I plan to use whole genome sequencing to identify certain characteristics about the STEC isolates we have, such as the presence and prevalence of virulence genes and serogroups. And I'll also be doing 16S RNA sequencing to identify changes in the microbiome and see if these correlate to differences in foodborne pathogen shedding. I'm currently still working on this project, but we already had some preliminary data to work with. So in table 1, we can see that the prevalence of STEC varied mostly by the treatment sampling day and the year they were taken. And then in table 2, we can see that the virulence gene STX1 only was found to be the most common, while it was less common to find STX2 only and STX1 with STX2 together. So based on this data, I predict that we will be seeing grazing influence changes in the microbiome and that there will be a higher prevalence of foodborne pathogen shedding in sheep post-grazing. And finally, just to talk about some future directions for this work, it would be very useful to also evaluate the prevalence of STEC in soil and produce samples in this mixed crop livestock system so that we can see if contamination is occurring after grazing events. And we can also evaluate other factors that could contribute to changes in the livestock microbiome, such as age and environmental conditions. Thank you for watching.